RNA interference by Sidra Aziz. Some background information about RNA interference. RNA interference, or RNAi, is a technique used to silence genes and regulate gene expression in many organisms. It is the same concept found in plants and fungi, a co-suppression in plants and quelling in fungi. RNAi involving small interfering RNAs, siRNAs, and microRNAs, which is miRNAs, through these steps of gene expression can silence specific mRNAs in the cytoplasm. An application that describes this technique. So the first step involves degradation of the double-stranded RNA into small interfering RNA, which is siRNA, 21 to 25 nucleotides long, by an RNase 3-like activity. In the second step, the siRNAs join an RNase complex, RISC, which is RNA-induced silencing complex, which acts on the cognate mRNA and degrades it. Several key components such as dicer, RNA-dependent RNA polymerase, helicases, and double-stranded RNA endonucleases have been identified in different organisms for their roles in RNAi. Some of these components also control the development of many organisms by processing many non-coding RNAs, called microRNAs. The biogenesis and function of microRNAs resemble RNAi activities to a large extent. Recent studies indicate that in the context of RNAi, the genome also undergoes alterations in the form of DNA methylation, heterochromatin formation, and programmed DNA elimination. As a result of these changes, the silencing effect of gene function is exercised as tightly as possible. Because of its exquisite specificity and efficiency, RNAi is being considered as an important tool not only for functional genomics, but also for gene-specific therapeutic activities that target the mRNAs of disease-related genes. Figure 1 diagrammatically illustrates the systematic spread of RNAi in plants. Such recovery occurred by a PTGS-like mechanism, because 19S and 35S RNA encoded by the cauliflower mosaic virus were degraded while cauliflower mosaic virus DNA was still replicating in the nucleus. Induction of the PTGS was visualized if the cauliflower mosaic virus infection and subsequent recovery were followed up in a transgenic bNAP expressing a P35S Gus beta-glucuronidase transgene. At the site of inoculation, Gus silencing associated with local lesions were first observed 7 days post-inoculation. Gus silencing eventually spread systematically, and the Gus activity of the entire plant was suppressed by 50 days post-inoculation. In this particular example, cauliflower mosaic virus acted as the inducer of the PTGS for the transgenes, sharing homology with the virus within the transcribed region. However, the virus itself was also the target of the induced PTGS, since 19S and 35S RNAs were found degraded. Here, this schematic illustration of systematic viral spread as well as RNAi and subsequent viral recovery in plants. The green and red indicate the presence and loss of GFP fluorescence, respectively, and orange denotes the presence of both colors. The red dots on leaves show viral lesions, and the bold arrow indicate the stages of plant growth, and the leaves are numbered accordingly. An arrow with the thin line shows a newly emerged leaf recovered from viral attack. The question I want to ask, how have cancer researchers used this technique to suppress a gene that is activating on cancer cells? Maria Francis?